Wow. Okay. Good morning, good afternoon, good evening, everyone, wherever you are. So I'm happy to meet you again this morning. It is really my great pleasure to welcome you to today's webinar on the cryopreservation of clonal crop and recalcitrant seed. So we are delighted to have such a diverse audience joining us. I see that we are already 44. I hope the other will join while we are being progress in the program of for what promised to be a highly insightful and engaging session. So this webinar will last approximately 1 hour and 30 minutes, including the time for question and answer at the end of the two presentations. So to ensure a smooth experience for all, we kindly ask you to keep your microphone mute, please, during the presentations. The Q&A session will take place after both presentations, so that you can please hold your question up to the end of the both presentation. So without further delay, I will introduce you now. I have the honor to give the floor to our first presenter, who is Dr. Rainer, who is a scientist at CIP, Genebanks. So Rainer, no, it's Bart, sorry, it's Bart. Bart is the first presenter. Who is the,
yeah,
Dr. Bart is the first presenter, and Dr. Bart is a Genebanks manager at Alliance Bioversity CIAT, based in Leuven in Belgium. Dr. Bart, you have the floor. Thank you. And sorry for the confusion. Thank
no
you.
problem. Okay, thank you, Marie-Noël. Hello, everybody. Welcome to this second week of seminars. So together with Rainer from SIP, we decided to split our presentations in two, or to give two presentations, one on an introduction on cryopreservation, what it is all about, and then Rainer, within half an hour, will present something more on the applications of cryopreservation. So I'm trying to share my screen. Okay, let's see. Okay, you see my screen?
Yes, I can see it very well.
Okay, thank you. So it's all about an introduction to plant cryopreservation. Before, it's always nice when you deal with such technology that you know the basics of it. So, okay, it's not moving. Okay, it is. So what is cryopreservation? So cryopreservation is a process where you store biological material at ultra low temperatures. So most of the time, the temperature of minus 196 degrees Celsius is taken as storage temperature. There are two reasons for that. The first reason is that we know that at this temperature, all biological, chemical, and physical processes are arrested. So whether you store something in liquid nitrogen for 1 hour or for 100 years, it should remain the same. And secondly, it's also practical because you can store material by plunging it into liquid nitrogen, so in big dewars or dewer tanks. But of course, it's not so easy. It's not just plunging things in liquid nitrogen and hoping something will survive. That is, of course, not the case, because when you expose something to such low temperatures, many things can go wrong. So you have different effects of this low temperature. So you have the temperature itself. So low temperature have effect on membrane stability. You have mechanical effects of extracellular ice crystals. You have dehydration-related effects. But what is the most important is the effect due to intracellular ice crystal formation. Why? Because such ice crystal, when they occur inside a cell, will result in penetration of membranes, so the semi-permeability is lost, and so this has to be avoided. So this is the trick. If you want to do cryopreservation, you have to avoid the formation of intracellular ice crystals. How can you do that? So you could say, okay, let's remove all the water from the cells, so there is no crystallization taking place. But of course, this is also not an option, because this way you will also kill all the cells, because cells, water has also a structural task in cells. So when you remove all the water, you will also kill the cells. So this means that you still need water in the cells and have no ice crystals when you expose them at ultra low temperatures. And the only way to do that is using a phase. Maybe you remember this from previous courses. The glass phase or the vitrification phase. So what is vitrification? It is the solidification of a liquid without formation of ice crystals. I have shown here an hypothetical ice diagram, or but maybe to go to explain you a bit more, I use the following slide. So you see here on the left, number A, this is water. Water, you know, these are molecules which are moving in a chaotic way, and your water is liquid. When you decrease the temperature, you have the formation of ice crystals. So your molecules will organize themselves into a crystal. And this has practical consequences, because if you look at figure D, there you see a biological system. So below you see the cell content, above you have the extracellular solution, and there you have the membrane bilayer, a lipid bilayer. And you see when a crystal is forming, your bilayer will be crushed, will be damaged, and you see the semi-permeability is lost. In case, however, you can make sure that you do not have ice crystals by use of vitrification. So this is a vitrified state, number C. So in fact, you have the same chaotic organization of molecules, but with that main difference with A, that the molecules are not moving. So you suddenly stop the movement of the molecules. So this way you have a solid, water or solid liquid, but without the formation of ice crystals. But very important is that you do not let them organize into a crystal. And this has consequences. In figure F, you see that indeed your membrane remains intact. So this is how it looks like in reality. Liquid, it's completely transparent. Water, which has crystallized, it's like snow. And then you have water that has vitrified, it's also transparent. So how can you have this vitrified solution? In fact, there are two requirements. The first one is that you concentrate your cellular solution. In fact, what you do is you make sure that there are a lot of molecules in between the water crystals, so the water molecules. So the water molecules have more difficulties to organize themselves into a crystal. So that is one thing. And the second thing is that you apply rapid cooling and thawing rates. Rapid cooling rates means that you do not give the time of the water molecules to organize themselves into a crystal, because you go from liquid to solid in a few seconds. The ice crystals do not have the chance to be formed. So in fact, these are two requirements for a liquid to vitrify. So the first requirement, concentration of cellular solution. How can you do that? So one way to do that is to use just air drying, evaporation. I suppose you have an hypothetical cell, you expose it to air, water is leaving the cells, is evaporating. The result is a concentrated cell or concentrated tissue that when you plunge it into liquid nitrogen, fast cooling, you get you avoid the formation of ice crystals. How can you do this? This dehydration or this evaporation, exposing the tissues to the sterile air of a laminar airflow cabinet, or in case of seeds or embryos, often dry silica gel in a closed container is used. A desiccator, it's also called a desiccator. Second method to concentrate your cell solution is using slow freezing. So this might sound a bit contradictory to what I said before. You have to use fast freezing rates, but this slow freezing rate is in fact there to dehydrate your cells. So I will explain. So again, this is your cell, your hydrated cell. You expose this cell or this tissue to gradually decreasing temperature, and this can be, for example, 1 degree Celsius per minute drop. And so first, crystals are formed outside the cells, extracellularly. Why? Because the osmotic content of the extracellular cell water is lower than inside the cells. So here, ice crystals will be formed. But it is because only water freezes and forms ice crystals. The remaining liquid is concentrating. And because intra and extracellular solution are in osmotic equilibrium, water will go out of the cells and will make sure that these ice crystals becoming bigger and bigger. So this is what we call freeze dehydration. The more you lower the temperature, the more water is leaving the cells. And finally, at, for example, minus 30 or minus 50 degrees, slow freezing, you have a concentrated cell that when you plunge this in liquid nitrogen will vitrify and not form ice crystals. So how can you do that? You can have computer-driven devices. You have stirred methanol baths. You have Mr. Frosty. It's a container you put in where you put the tubes and you put it in a minus 80 freezer, and then the lowering of the temperature is around 1 degree Celsius per minute. So there are different ways of doing that. A third way of concentrate your cell solution is osmotic dehydration. So here you use big molecules, sugar alcohols, which remain outside the cells, and by means of osmosis, water will leave the cells. So for this, you can use sugars, polyethylene glycol, ethylene glycol at low temperature. So again, leading to a more concentrated cell. Then you have the penetrating cryoprotective solutions, which are smaller molecules. A very well known molecule that is used often in cryopreservation is dimethyl sulfoxide, but you can also use glycerol, amino acids, ethylene glycol at higher temperature, and again, leads to a concentrated cell. And finally, and this is typical for plants, so the previous four methods can be used in all biological systems, so also in medical science. Here, it is typical for plants that in temperate regions, like in Belgium, where I'm located, plants are protecting themselves in autumn against winter temperatures, against freezing temperatures, and thereby they accumulate certain compounds like sugars, like polyamines, and leading also to a concentrated cell solution. So if you want to know more about this, we have recently published a paper in Annual Review of Plant Biology, where all these processes, in fact, are more described in detail. So you can go to that paper. So, but what is now the problem is that most hydrated tissues do not withstand this dehydration, which means that you need to protect them. and you can protect them by adding some substances that are protecting all kinds of organelles, membranes, and these sugars, glycerol, and so they are replacing the water, making sure that the structure of the membrane remains. But you also have adaptive metabolism, where also cryoprotective substances are induced. So this is the theory behind the cryopreservation. So how is this cryoprotection working? And this leads to the development of different protocols. So I will quickly go through the different protocols. So what protocols are used? I will not go into detail, but just give you an idea of what is there on the market, in fact. So, in fact, maybe we can first distinguish the different cryopreservation methodologies in two sections. The first is the dormant bud cryopreservation and some air drying, which means that this is material that is not relying on in vitro culture. So these protocols, you do not need laminar flow benches, you do not need autoclaves, you do not need in vitro culture. So the first well-known example is the dormant bud cryopreservation. So what is that? So in fact, you isolate dormant buds during winter in temperate regions. You expose them to, so they are exposed to cold. Then you do some dehydration at the air and also freeze dehydration. And then you plunge them in liquid nitrogen for storage. So you need to optimize some parameters. But what is now the major problem with this kind of methodologies is that, first of all, it's only applicable to species producing dormant buds. So typically for often trees in temperate regions. And also the species need to be grafted, because after the cryopreservation of these dormant buds, you need to regrow them. And of course, an individual dormant bud, you cannot let it root just by putting it into soil. No, you need to graft it. And so this is why these are the two requirements to apply this protocol. So this is how it looks like. For example, this is how you cryopreserve dormant buds from apple. You go to the field in winter, you excise the buds, the nodal sections, you slow cool them, dehydrate them, slow cool them, plunge in liquid nitrogen, and after storage for years, you can take the buds and graft them onto a root. Okay, so the second method is air drying. It's a very simple method, as explained already. We apply it, for example, for the cryopreservation of seeds of banana, but you can also apply it to pollen, zygotic embryos, and other orthodox seeds. So this is just air dehydration and storage in liquid nitrogen. So it's simple, but not applicable to many tissues, because you need to be able to dehydrate your until moisture contents of around 10%, and most of hydrated tissues do not withstand that. The slow classical freezing, I explained already a bit on that. So this is relying on freeze dehydration. So this was developed in the 70s, 80s. So what you do is you take cells or tissues, you treat them with cryoprotective agents. Often dimethyl sulfoxide is used there for the slow freezing methodology. And then you apply a freeze dehydration method. So again, here, quite some parameters need to be optimized. So, and this is only applicable to in vitro culture, in vitro tissues. And so then you need to cool down. And what is important in this cooling process is that you apply a cooling rate that is not too fast, because too fast will result in too little dehydration, or not too slow, where it will result in too much dehydration. And both of these things can result in that. So this means that you need to find somewhere a freezing rate, which is mostly around 1 degree Celsius per minute. It is often applied to cell suspensions and calyces because it's a methodology which was used in the past for all kinds of tissue, but now it's mainly used for unorganized tissues like these calyces and cell suspensions. Then we have encapsulation dehydration method, so where in fact you take a meristem, or this can be an embryo, you encapsulate it in a small alginate bead. And then this alginate bead is treated with high sucrose concentration. So this is the osmotic dehydration. Then followed by air drying. And then this is followed by rapid freezing. Again, you need to optimize a lot of parameters. But what is the main disadvantage, according to me, of this method is that there are a lot of steps involved. You need to encapsulate. So in fact, practically, it can take up to 1 week, starting from your starting material until something is really in the liquid nitrogen. So it is very labor intensive and not always efficient. But it's still being used. Then we have the doppelt freezing. I'm not going to go in detail there because this is only used for potato shoots. Pre-culture fast freezing, I will not also go into detail because it also has limited applications. And then the vitrification, this is an important one. In fact, in 1990, Professor Sakai, he was one of the pioneers of cryopreservation. He developed a vitrification protocol relying on PVS2, plant vitrification solution number 2. And this is a solution which is a bit syrup-like, a very highly concentrated solution. But he found out that when you expose meristems or embryos to this solution for times ranging between 10 minutes and 2 hours, that you can have vitrification of your cell content even when applying fast freezing. So this was a real revolution because it's a simple methodology, and later on we have proven that it can be applicable to all kinds of tissues. So this is really one of the bases of the plant cryopreservation for the moment. But of course, again, you need to optimize a lot of parameters. And with this methodology, it's not so much the cooling rate that is important because you do extremely fast cooling. But what is more important is the exposure time to this vitrification solution. A too short exposure time will lead to not enough dehydration, meaning that you will have crystal formation when you plunge it in liquid nitrogen. And too long exposure to this PVS2 solution will result in toxicity. So it's an important parameter here that needs to be optimized. Encapsulation vitrification, I will also not go into detail. And then we have droplet vitrification, which is for the moment, in fact, the protocol which is in Genebanks the most widely used. In fact, it is a combination of vitrification, so with PVS2 that I explained a bit before, in combination with the application of ultra-fast freezing and ultra-fast warming rates. So I explained already in the beginning of my talk that this ultra-fast freezing is also ultra important to avoid this ice crystal formation. So the normal vitrification protocol that was developed by Professor Sakai used cryotubes. So you see here a 2 milliliter cryotube. You put some meristems in the cryotube. You add an extra 1 ml of vitrification solution, and then you close the cryotube and plunge the cryotube in liquid nitrogen. This results in a cooling rate of about 6 degrees Celsius per minute. Why? Because it's a volume. It's a 1 square centimeter volume, and you can imagine that the central part of this volume will have a slower freezing rate than the external part. So this is why when you use smaller volumes, you can use this will also result in much faster freezing rates. And this is how we developed it. So what we do, instead of enclosing the meristems or your tissue that you want to cryopreserve in a cryotube, we put it on a piece of aluminium foil and we plunge the aluminium foil in liquid nitrogen. And the freezing rate or the cooling rate there is about 130 degrees Celsius per minute. So this means that in 2 seconds, you could go from the temperature or room temperature until the temperature of liquid nitrogen. And this is something which makes a huge difference. Something intermediate are the straws, so where you can place the meristems inside a straw, but this is now less used anymore. So now this is the methodology which is often used. So this is how such a typical protocol looks like. You have a plant, in this case, this is dragon fruit, for example. It's a kind of small cactus. Excise meristems, treat them with loading solution, treat them with PVS2 solution, put them on a piece of aluminium foil, plunge the aluminium foil in liquid nitrogen, then put the aluminium foil in a cryotube for storage, and then for recovery, we take out the piece of aluminium foil, we rinse it into a recovery solution, and we can regenerate a plant from the meristems. In the best case, of course, because it's not always working. And then we have 2 other methodologies, the V-cryoplate and the D-cryoplate procedure, which I'm also not going into detail because these are relatively recent protocols. And yeah, so the application is still less. If you want to know more about these cryopreservation protocols, I can also suggest you to read the paper, because also the cryopreservation protocols are described in there. So why are we using now cryopreservation? Of course, for the storage of genetic resources. Storage of genetic resources, Genebanks. You have in situ versus ex situ conservation. You have, like most of the collection, have seed collections, the seed preservation. And the cryopreservation we are going to use in case seed preservation is not possible. So in which cases seed preservation is not possible? It's in case, of course, when you are dealing with seedless crops, like bananas. Edible bananas do not produce seeds, so you have to look for alternatives. Then you have those crops or trees that are not breeding true from seeds, like the fruit trees, apples, also other fruits. You need to preserve the clone, because this is the genetic combination that you store, and when you make seeds out of it, you lose this specific combination. And then finally, of course, you have crops with recalcitrant or short-lived seeds, where also normal seed preservation is not possible. So in this case, cryopreservation is a good solution. There are also some other applications of cryopreservation. I will not go into detail, I will just mention them. So cryopreservation can also be used for the storage of specific cell lines. For example, in case of banana, we have 1,000 tubes containing embryogenic cell suspensions of banana stored in liquid nitrogen. So this is in fact to store material that can be used in gene editing. We can use also cryopreservation to eradicate viruses, and this is called then cryotherapy. It can also be applied as a kind of breeding tool. For example, in conifers, you do some crossing, some hybridization, and these small embryos can be then cryopreserved, and later on, when it seems that this specific cell line is a good one, you could cryopreserve it. And then finally, also commercially, cryopreservation can be used to store clean stock cultures in production labs. So to conclude, my presentation is, I think it's clear that cryopreservation is all about preventing intracellular ice crystal formation during cooling, as well as rewarming, because this is also important. Also during rewarming, the crystal formation cannot take place. There are different cryopreservation protocols developed and available. Which one to use depends on different parameters, the species, tropical versus temperate, monocot versus dicot, woody versus herbaceous. So, for example, in case you can use dormant buds, use the dormant buds because you are not relying on in vitro culture, but this is only possible for certain species. Also, the tissue type plays an important role, whether you are dealing with seeds, embryos, calli, cell suspension, dormant buds, shoot tips, pollen. This all has its influence of which protocol to use. And also, local conditions can also have an influence. Do you have experience with a certain methodology? Do you have equipment? Do you have equipment for slow freezing, for example? And then finally, so it can be used for storage of genetic resources, but also for eradication of viruses and other microorganisms, eradication of bacteria as a breeding tool and as a commercial stock deposit. Okay, this ends my presentation. Of course, this work can never be done without the collaboration of many different institutes, many partners, and which you see here, also my collaborators in the lab. And thanks for your attention. And I think now it's time for Rainer to give some practical applications of cryopreservation. Thank you.
Thank you, Bart, for this excellent presentation. So it will be easy for me to go on. I will share now my screen. Can you see the presentation?
Yes, perfect.
Okay. So my name is Rainer Vollmer. I'm working at the International Potato Center, and I'm in charge of the cryopreservation program there. Okay. Well, let's start with the beginning. What are clonal crops? We all know this, but it's good to remember how important they are. Here you have a definition. There are different ways to say this. Clonal crops are cultivated plants that are propagated vegetatively rather than from seeds, ensuring uniform genetic characteristics across the entire crop. There are some examples, and this method allows farmers to maintain desirable traits like disease resistance, yield, and quality consistently across generations. And it's really surprising that 5 of the 10 most important crops that are most frequently consumed by humans, are clonal crops. You have them here below on the bottom. But there are many more, and surely you will find there some that you have in your region. And we could fill some more slides. There's only one here, but you can see, there are many important. There are also many other fruit trees, forest trees. Here have a short overview of what are clonal crops. This a little bit, also Bart mentioned this, for clonal crops we have 2 different conservation methods. One is in situ and the other is ex situ, and then some hybrid approach. You can see in situ is on farm conservation. The hybrid approach is community seed banks. And then within the ex situ section, you have field banks, in vitro, cryopreservation, and also DNA banks and botanical gardens. The most frequently used at present are surely in vitro conservation and also cryopreservation. So therefore, we will take a little bit a deeper look at these at three of them, also considering field collections. What are the advantages and disadvantages of each of them? For in vitro, of course, the material is maintained disease-free. It's relatively space efficient, and you have always access to the material. But then you have the disadvantages that is very labor intensive to maintain the collection. You have the risk of contamination and also of somoclonal variation. For cryopreservation, the main advantage is, of course, long-term conservation. Low maintenance cost, you do it only once, if you do it right. So it's very cost effective on the long term. But on the other side, it's complex, as you have also seen from Bart's presentation. You need a high initial investment, and also you have recovery challenges. You have to develop a protocol for each crop. It's not something that works for all of them. There's no golden solution. Field collections, yeah, you have it under natural growth conditions. It's easy to evaluate. You have you can multiply it up on a large scale and use it directly. But the big disadvantage is it's highly vulnerable. It's very cost intensive, and also the access is a problem. And especially in times of climate change and pest growth and appearances, maybe field collections is not the right way to go for ex situ. What's this? This is the reality, a little bit of Genebanks. It's sad, but it's the reality. You have their collection, let's say 100%. Of this collection, how much is really characterized that you know, is it's phenotyped and documented? It's about 50 to 30%. And how much is used? 10 to 20% in average. And for active breeding, 2 to 8%. This means that passively conserved, as it's only maintained over time without doing anything else, you have a huge portion of 70 to 85%. So how to deal with this for clonal crop? And therefore I would like to present you a short exercise that we would like to go through with you. Let's consider the following condition. Yeah, we have a clonal crop in vitro, and we assume a probability for some negative events that could occur during a multiplication cycle. Of course, there's human mistake, there's mislabeling, and somoclonal variation. It's only 3, this could be much more, so it's only an example. So if you calculate the probability that one of these 3 occur, this would be 3% in one multiplication cycle. But when you do 10 multiplication cycles, then the probability of occurring one of these events would increase to 26%. And if you do 30 of them, then you have 51% or 52%. So this 52% may correspond to 10 years of conservation, 20 years of conservation. So under the line, at some point, these things will occur, will happen. And we only considered 3 of the possible negative effects. No? There are many others, and we don't know the probability of them, and surely it varies between crops and organizations. But this is the big disadvantage of maintaining a collection clonally continuously through propagation and multiplication. And then there we have a comparison of in vitro conservation versus cryoconservation. As the conservation period for in vitro varies, can be as short as 3 months, but also you can, through cold storage, you can increase it for some crops to up to 36 months. While for cryopreservation, there's no time limit. You can conserve it for decades to centuries. The cost to conserve one accession for 1 year in vitro under our conditions is $120. And for cryo, also under the conditions of where I'm working, is $500. an initial one-off cost, as it's a unique cost, but then year per year it's only $7 to $10. This means that after 7 years of conservation, cryopreservation is cheaper than in vitro conservation, but this depends on the place where you're doing cryo. No? It's only an example. Then also you maintain more plants in cryo. In vitro we conserve in 3 to 6 test tubes per accessions. So we conserve about 12 plants, while in cryo, in our case, we conserve 120. Of course, we will not recover all of them, but if you recover, let's say, if you have a recovery rate of 50%, you still get 60 plants. No, it's 5 times more than in vitro. The process capacity per person in an 8-hour shift for in vitro is 720 plants, more or less, if you work with different genotypes. And for cryopreservation, it's of course less, but still very high in the case of potato for 450 shoot tips. But again, as also Bart mentions, this varies between crops, as it's a different thing to excise a shoot tip of potato shoot tip than, for example, a banana meristem. It's much more complex to excise it and takes much more time. No? Space requirements. So, well, for in vitro, we know we need a room with a light system and air conditioning system, filter system. And for cryo, the conserving medium is the liquid nitrogen. So we only need three tanks to conserve about 5,000 accessions. So then comes the questions why cryopreservation. Only complementing some things Bart already mentioned. So human science is really pushing this forward. Cryopreservation, more and more cell types, tissue types are conserved. And also this means we apply cryopreservation for our sons, no? So we rely on it for our life, no? So it's not, it's really proven to work in biology. Then, of course, there's no time limitation of conservation. The material stays genetically stable. And then the reduced labor needs. This was very, very impressive when we have gone through the COVID times. So it was enough that one person came in, filled up the tanks that contained the samples with liquid nitrogen every 3 weeks, and one single person could conserve this way thousands of accessions. No? While to maintain an in vitro collection or other field collection under these extreme conditions of COVID was horrible. No? And we many of us know this was really hard work. So the other thing is you don't need a lot of space. We already mentioned this, that the cost is lower than in vitro after 7 years of conservation under the conditions we work. It's also highly resilient not only to COVID but also to disasters, because you have the material within a stainless steel tank and within boxes, and then within the boxes in small vials. So it's physically very well protected, and it also, as it has a low cost, conservation cost, is that if there's a budget cut, it will not be difficult to maintain a cryo collection because it's cheap. And then there's a new potential application that will get more important if a production system goes down, and because of a disaster, for example, or extreme drought or whatever, and you need to build it up quickly, you will need thousands and thousands of plants in a very short time period. But to get them, you need many multiplication cycles generally, because you don't know when it will happen, and nobody will maintain thousands of plants only to maintaining them of one single genotype. So, but in cryo, this is possible. You can maintain, let's say, 10,000 plants of a certain important variety in the cryo tank, and if you need it, you can quickly thaw it, and with one multiplication cycle, you can bring it to the greenhouse. For the clonal crops, what are the most frequently used explants for cryopreservation? It's principally three. The most frequently used are surely shoot tips. Then we also have the axillary buds, and there are also some experience with nodes, but not so successful. And then you have the dormant buds that Bart has already explained. This is perhaps these are the three most frequently used methods for conserving clonal crops, for cryopreserving clonal crops. We have the B plate, the D plate, and then the droplet vitrification. The B plate and the D plate, that's a little bit you stick the shoot tips with alginate on plates, and then you move these plates through the process, while for the droplet vitrification you use directly aluminum foilers. The advantage is that the aluminum foil you will always have easily accessible wherever in the world, but to produce this cryo plates, this might be a challenge, because they have exact dimensions and it's not so easy to obtain in everywhere. But both methods work very well, and at the end the important thing is that you get complete normal looking plants when you rewarm them, whatever is the method you use. For clonal crops, frequently there's a pre-culture step, while for template crops, for some of them at least, cold acclimatization works fine before you dehydrate the cells and vitrify them. This is only one example for protocol that works for potato for us very well. So we use in vitro plants that we multiply at 7 Celsius. We multiply in deep petri dishes, and we let them there for one week that the bud breaks, and then we place them at 7 Celsius for 2 weeks. So then when the plants have 3 weeks, we excise the shoot tips. It's about more or less 1 millimeter, and place them on small filter papers. You can see it in the lower photo. So each filter paper contains 10 shoot tips. And then the cryoprotection with the PVS2 solution. First, the loading solution, LS, 20 minutes, that protects the shoot tips against the phytotoxicity of the second solution, of the plant vitrification solution 2. In this case, 50 minutes, as Bart also has explained. These times vary from crop to crop, are different. And then in our case, we absorb this shoot tips with a Pasteur pipette after the chemical treatment, and place them within a droplet of PVS2 on a small aluminum foil strip. This is, you can see it in the lower photo of the letter D. And this is then quickly plunged into liquid nitrogen as a rapid cooling. And then we store it in small tanks on aluminum canes. But of course you have to check if it was successful. So you do a viability testing. In our case, we rewarm 30 of 150 shoot tips in rewarming solution for 20 minutes. And then we recover it first in darkness, very important to reduce oxidative stress, and then normal light conditions. And then we do the assessment of the recovery. We do it twice at 30 days, and then after 50 to 60 days. And if you get a minimum, in our case, if you get a minimum regrowth rate of 30%, then we decide to transfer the samples to the cryobank. If not, we throw it away. We only want to store material in our cryobank. We are sure that we recover complete normal looking plants with a good recovery rate. This is the same thing, only summarized. So in vitro propagation, cold acclimatization, shoot tip excision, treatment with cryoprotectant solutions, and then place it in a droplet of PBS2, plunge it into liquid nitrogen, and store it in the cryo vials, and then transfer it to a small cryo tank. Then do the viability test, and depending on the recovery rate you get, you take the decision to transfer it to the cryobank or not. This is very important. The only thing that matters is on the left. The right are nice pictures perhaps, but it doesn't mean anything. It's only survival. All decisions should be taken depending on the full plant recovery rate you get. If you have a cell phone, you can read this QR code. So we have prepared a detailed video of the cryopreservation process that we apply. There's an English version and a Spanish version it shows the whole process, many details, all the chemical compositions and of the media, of the solutions. So it's a useful, let's say, reference, because sometimes it's better to see it on paper. You can write many things, but there are sometimes small little things that are important to know, and we think this video might be useful if you're interested in cryopreservation. So please feel free to read this QR code and get the link. Then where did this start a little bit to that because in recent years we really scaled it up a little bit, cryopreservation. This started with a feasibility study that was done in the year 2017. This was also, for example, Bart was participating very deeply in this study, and thanks Bart for sharing some slides on this, so I can show it here. This study was done 2017, but then they made a second survey in the year 2021 and more institutions responded to this. And I would like to share with you some of the results of this feasibility study. Again, you have their QR code in the middle. So if you want to access to this feasibility study, please read it and store the link. As you can see, they contributed many CGIAR centers as Bioversity, SIP, but also the Crop Trust and the German Development Service, and also from Australia and Switzerland development services. This is also what was shown as collaborators in Bart's presentation. So what were the conclusions of this study? The principal two conclusions were two. A major global initiative should be launched to accelerate the development and implementation of crop cryopreservation for important crops. Also there were estimated there are about 100,000 accessions that should be cryopreserved. And then that cryopreservation should be used as a safety copy, safety copy options for clonal crops, similar as Svalbard is for seeds. There's the institutions that participated and responded to the survey. As you can see, there's some international centers, but also highly specialized centers from the developed world, from Europe, and also other national centers from Latin America, from Europe. And this is what was seen, as in 2017 in the first survey 9 crops were cryopreserved with more than 100 accessions per crops, but this number increased to 17 crops for 2021. So we made progress, and that's nice to see. As you can see, the most frequently cryopreserved crops are potato, banana, cassava, apple, garlic, and then others. And this is, if we split it up by type of explant, you can see the shoot tips are the most frequently used explant type, followed by dormant buds. And on the right side, you see it for some crops. So for some crops, we exclusively shoot tips are used, as for example, banana, potato, sweet potato, also very frequently for strawberry and rubus. But then for others, dormant buds like apple and mulberry, and also coffee, for example, seeds, citrus, many embryos, as it depends. Then, based on this, we move forward, and in 2022 we had a workshop, a big workshop in Latin America, with the participation of 14 countries, and 10 of these 14 countries have received an intensive 2 weeks training on cryopreservation, going through the whole process, and 5 of them already started to implement their cryo programs. We are helping them, we are supporting them where we can. So the idea is to really scale up cryopreservation, not only in Latin America and Caribbean, all around the world. It's in Africa, Asia, Pacific, Europe, wherever. I think cryopreservation is the future of conservation of clonal crops. Then this will happen 2022. Then 2024 in November at the International Potato Center, we inaugurated a new cryo vault, a new cryo bank that has much more space for our collection, but also for 70,000 accessions of other institutes. So this is why we offer the option that they can deposit safety copies, especially from the region, there in our cryo bank. You can see there's some cryo tanks in the building. And then the numbers of our cryo bank, as you can see, we have a lot of material already in cryopreservation, especially potato, but also sweet potato is growing. This happened November, and 1 month later, then we had the CGIAR cryo workshop, where 7 CGIAR centers gathered together and shared their experience, showed their protocols. It was very nice, as we learned a lot from each other, and I think we can build on this and really bring it to the next level. And finally, this happened recently, last month. There were published 2 new practical guidelines for the application of the Genebanks standards for plant genetic resources from the FAO. So one is on cryopreservation on the left. You can again scan the QR code, so it brings you directly to the page to download the document. It's there free, of course. And then also on conservation of species producing non-orthodox seeds. This is also recently published. There's valuable information inside there. So if you want to deepen some things, it's surely a good source also. So that's it from my side. The final message would be, if you're working at the Genebank or close to a Genebank and you're interested in cryopreservation, please feel free to contact us so we can discuss potential trainings, support for implementation, technical guiding, whatever. So here you have the email of Bart and my email, and thank you very much for your attention. So
thank you very much. Thank you very much. Thank you so much for the excellent presentation from Bart and for Rainer. Thank you very much for your contribution to this webinar series. Now, the floor is open for our participants to raise questions, make suggestions or comment. So please, to raise questions, you can raise your hand. You have the icon with the hands. You can use it to raise your hand, or you can write in the chat. I hope the chat is open for participants to write. So the first question is from Ahmad. Please, Ahmad, unmute and then raise your question. Thank you. Ahmad, can you hear me? Hello? Can you ask your question, please? You have your hand is up. Or is it a mistake? Can you speak? No, you cannot speak. Okay, I don't know what is happening. I want to see if you have other question.
Hello, can you hear me now?
Yes, we can hear you now. Go ahead.
Oh, thank you very much. Okay, so first of all, thank you very much for such a wonderful presentations, starting from the basics of cryopreservation to the advanced level. I have a couple of questions, and the questions are probably linked with each other. So my first question is, what the experts think that what could be the potential epigenetic changes induced by the cryopreservation in plant genetic resources?
Bart, do you want to?
Yeah, go ahead, Rainer.
Okay. Oh, yeah, I maybe Rainer can complete that. Okay, so this is a question that often arises, of course. It is known that in vitro culture, especially when in vitro cultures are under stress, that can give rise to somacal variation. Somacal variation means mutations that are arising from the in vitro culture itself. Why? Because clonal material, in contradiction to seed material, and every time a seed is made, there is a kind of repair mechanism. Yeah, in case of clonal propagation, mutations accumulate. But this happens in the field as well as in vitro. So the question whether on what is the effect of cryopreservation of somacal variation? Okay, so the somacal variation could eventually arise during the preparative phase, the in vitro culture phase, but during the storage itself, then the risk for somacal variation is almost nil, because nothing is happening. The DNA is fixed, everything is fixed. There's no oxidation, there are no Rosses coming. So during the storage, we are quite safe with respect to mutations, but it are the preparatory step, which also take place during in vitro culture, that can give rise to somacal variation. So maybe Rainer, you want to say something extra on that?
Yeah, so maybe to identify somaclonal variation, it's very difficult now. So you would need full sequencing probably to really find them. And there are very few studies out on this because it's still expensive and takes a lot of work also on the bioinformatic level. And then there also you have epigenetic variations that sometimes people see something and might interpret it the wrong way. So it can also happen on methylation of the DNA. So you will see their change, but it's not a somoclonal variation.
Thank you very much. So,
Okay.
my next, then it is linked with this question that, okay, then how can we um, uh, monitor the stability of any genetics of any crop or the plant genetic resources that we have conserved using cryopreservation? How can we employ the omics based tools or any molecular tools to identify the stability of our germplasm that we have stored or preserved using cryopreservation technique? Thank you very much.
Yeah. Okay, I think that Rainer partially responded to the question already, but okay, there was a time that people or that researchers tried to look at using AFLPs, rapids to see for some collaboration to detect. So of course, this is not really a very valuable method because this is only screening even not 1% of your DNA. So like Rainer said, complete full genome sequencing, this is the only way to see whether you have point mutations or whatever. But then, like also Rainer said, if you are dealing with epigenetic methylation, your full sequencing is not going to detect that. So deep
Yeah.
proof is putting something in the field. That is deep proof, whether something is normal or not. But of course, full sequencing is a good thing. But yeah, for the rest, there are no easy solutions for that. No.
Perfect. Perfect. So I have seen Rainer have presented in his last slide, I think, or the second last slide, that most of the plant genetic resources preserved are the clonal propagated crops. I want to know what is the scope of cryopreservation in orthodox or recalcitrant crop species?
For recalcitrant seed species, yeah, because of course recalcitrant seed species you cannot store by drying in -20. You have to look for alternatives. So there are some research groups who are looking whether it's possible, for example, to cryopreserve the seeds by dehydration, but that's a difficult one, but especially by cryopreserving the isolated embryos or embryogenic axis. So this is something which is being researched now for orthodox recalcitrant. For orthodox species, I also see room for cryopreservation because, with orthodox seeds, even when it's orthodox, whether you can store it for 50 years, you still always need regeneration phases. And when you regenerate, there is always the risk of losing diversity. So I think that even for orthodox seed species, cryopreservation could be good to store a reference collection or a core collection. But this is my personal thinking of that. I think that this should be considered a reference or a core collection preserved in liquid nitrogen, and this for wheat, for maize, for all these things. But that's something to be investigated, I think.
Okay, thank
Perfect.
you.
Perfect.
So you
Thank
have
you very much.
okay, thank you. So we have one question in the chat. It's about they want to know the genetic stability of cryopreserved material. Who want to speak on that? And then they say that the stress due to low temperature can cause damage in DNA and also it can cause the genetic variation of the material which are cryopreserved. So this is a comment. So if you want to say something.
Well, I can start. Perhaps the first one we already answered somehow. So it's really very dense markers, ideally full genome sequencing that if you want to verify it. But the same question as Bart also mentioned applies for in vitro, for field collections, for anything. No? So this is just part of the game. No? The second one is if cold can cause DNA destruction. I would like to highlight their point that going out of leaving the plants behind, we rely on embryos and sperm cells for humans, for artificial insemination. So this is something we always should have in mind, so that we are really where human science is taking let's say is ahead of the game. We can be very, let's say, we can rely on this. No, it's not that we are doing here hocus pocus science. This was already confirmed that this works for humans, for animals, and for plants, and that cold at 7 Celsius may stress plants, but at minus 196, I don't see this. I don't know what do you think about this, Bart?
I was muted. Yeah, sorry. No, during cryo, I think that nothing much takes place. Yeah, it's and indeed it's like Rainer said. I mean, if it would, so sperms well are also biological materials. That the conditions are the same. Cryopreservation. If there would be a serious risk of abnormalities, I don't think that medical science would use cryopreservation for embryos or for sperm cells. They would not take the risk, I think. So I think we can be quite sure that the DNA is very well kept. Yeah.
Okay, thank you. So we have 2 more questions in the chat. We will finish with that before we give the floor to Madame Elizabeth to raise her hand. So the person is Manamela, who is saying, thank you very much for your informative presentation. My question is on the regeneration capacity of clonal crops. Have you tried full regeneration of the cryopreserved material like sweet potato or cassava in the field where you successfully harvested the tuber?
Yeah, sure. Maybe I can answer maybe about our cases in banana, and then maybe Rainer can talk about potatoes. In bananas, we have indeed regenerated full plants. We have harvested them. We have compared yields. They were exactly the same. And in fact, we are using this cryopreserved banana collection also to replace the original accession in vitro when that proves to be abnormal. So in fact, we use the cryopreserved material also as a safety backup for the cryo for the in vitro in case that goes wrong. So we have regenerated, and the cryo itself is not influencing abnormality of normality of plants in case of banana. But I guess it's the same for potato. Rainer, up to you.
Right. Many years ago, this was done by Anna Panta. Was there done a study in comparing yields, morphological descriptors, molecular assessment, and there are many publications out there that have done this in different crops.
Okay, so now we have the question from our colleague, where I am. So we have the question, it's a very long one from our colleague. The name is Ojukaya. I hope I'm pronouncing well your name. Sorry if it's not the case. Thank you for the informative and insightful presentation. Which CGIAR center serve as the gene bank, and can you share the approximate breakdown of in vitro conservation and cryopreservation, including the data on vitrification in each of them? Can you share the approximate breakdown of in vitro conservation and cryopreservation, including the data vitrification in each of them? And there is another question. Maybe I go up to the end and then we start responding. Also, you mentioned about the passive preservation of the genetic material accounting for around 80%. How can the efficient use of genetic material be promoted in the CGIAR gene bank? Is there something to do with the formulation of SMTA or extension of Annex 1 of the treaty? So who want to go? Who want to start?
Bart, do you want to
Yeah,
go?
okay, I can start maybe. Which centers serve as gene banks? I guess that the question is there, which of the centers are applying cryopreservation to their gene banks, and how much material is there? So in case of banana, for example, we have an in vitro collection of 1,700 accessions and 1,300, so 66% more or less, are cryopreserved as a backup of the in vitro collection. We have, of course, the biggest one in SIP. Maybe you can say something about that, how much materials, but you have shown it, I think, Rainer?
Also for potato, more or less 90% of the in vitro collection we have in cryo, but for sweet potato, only 13%.
Yeah. And then we have IATA, which also has some hundreds of accessions cryopreserved of yams, also of cassava. We have CIAT, who is also working on the cryopreservation of cassava. So these are the 4 major CG centers which are applying cryopreservation. I don't know whether that response is a response to the answer. With respect to the passive preservation of genetic material, okay, the question is there, why not more material is being used? Is that a question, what you think?
Can we just give the floor to him? Please, Ojukaya, please, can you raise your hand? You open the mic. Where can I identify? Yes, I will enable your mic so that you can speak. Can you speak, please, so that we address properly the query? Go ahead, please.
Thank you for giving me the floor, and I think my question is well received. How can we use further the genetic materials that are conserved in CGIAR gene banks? This is my question.
Okay. Yeah, so the material is can be requested for often online. Of course, this material is always associated with an SMTA, Standard Material Transfer Agreement, which includes some restrictions and not direct use. But this can change. And how another way of more using it is that we need to be able to characterize our collection also better, because it's only when the collections are better characterized that they also be better used. And this is something where we still have some work to do, I think. In bananas, but I guess this counts true for most crops within the CG centers.
Maybe
Now, you want to say something, Rainer?
a detail perhaps. Also, the CGIAR is still the system that more material distributes worldwide. It has a huge impact. And if you imagine, let's say a collection of 5,000 accessions, and then you distribute only 20%, these are still 1,000 accessions that are distributed now, also that are used of the diversity. I think the problem is even much more complicated in the national gene banks than in the international gene banks.
Okay, thank you very much. Now, I can give the floor now to Elizabeth, please. Elizabeth Johnson, let me open your mic. Why is it not working? Can you unmute, please?
Yes, good morning. Good morning, colleagues. Good morning. And I want to thank Bart and Rainer for wonderful presentations, both. My question is probably more on the practical side. In terms of Rainer, you said that you had or you would store materials that had a 30% recovery rate. That's after the process. Is there any way to increase that percentage of recovery rate? And what are, to ensure, I mean, if we are storing something, for 20 years down the line, we want to be sure that we actually could recover the plants when they're thawed. So what are some of the things we can do to improve that efficiency? Thank you.
Also, the number that I said, 30% is the minimum. In the case of potato, our average recovery rate of the 4,500 accessions is 73%. 72% exactly with the newest protocol. In the case of sweet potato, we are much lower. We have our average recovery rate of about 30%. But even then, imagine you have one cryo vial with 10 shoot tips and you have a recovery rate of 30%. Then you will get of this 10 shoot tips of one single vial, you will get 3 complete plants that then you can multiply up for in vitro culture. This is, it depends. No, some institutes have 30%, others 40%. I think, Bart, you have a probabilistically more a similar approach, but more. Yeah, I will go over to you that you can give perhaps your vision on this.
Yeah, a percentage is, of course, maybe not always a good measure. That's why we developed in collaboration with IRD in France probabilistic calculation. And in fact, our calculation is an experiment is good, is successful if at least one plant can be regenerated out of the cryopreserved material for 99%, you see. So, and this corresponds to, let's say, most of the time to 25 to 30% of regeneration capacity. But most of the time, we have over 50% or 60, 70, 80%. And then we have a probability of 99.99% that something can be regenerated out of the cryopreserved material. And we are quite confident that this percentage is not decreasing with time. This is also something, the cryopreservation, where you store something for 1 hour of 100 years, the percentage, the regeneration percentage will not decrease. So therefore, we are quite confident with such probabilistic calculations that we are doing well. And yes, indeed, sometimes here we have to live with relatively low regeneration. Relatively low, I mean 35%, because not all accessions belonging to one crop species respond the same to cryopreservation. Some of them are difficult and some of them are more easy. And out of practical reasons, we chose to use one protocol for all the species, for all the accessions within one species. This is out of practical reasons. Maybe we could increase the individual accessions, the regeneration rate, but then, yeah, we should optimize for all individual accessions. And yeah, this is, yeah, if you give us a few 10 research staff, then we can do that. But
I know,
Yeah.
time consuming. Thank you. Thank you for your answers.
Okay, thank you for the discussion. We have Hamad, you have you raise your hand. Is it the old one or the new one, please? Can you
Yes,
unmute?
yes, it is a new one.
Okay,
Thank you
go
very
ahead.
much. My question is related to viability. In case of cold storage rooms and the old conservation practices, or the standard conservation practices, we can say, we often refresh our seeds, test their viability with after a certain period of time. Uh, my question is, in case of cryopreservation, what do you think? How long the genetic resources be stored, and after how much time we need to check their viability? Probably this may also be linked with the Elizabeth question earlier. She asked, maybe, I mean, the efficiency, whether the efficiency is linked with the time conserved or preserved the genetic resources or not. Uh, am am I able to deliver my question clearly?
Yes,
Okay, thank you.
I could answer something from our side on the first question. We have set up an experiment where we that includes the core collections and many other diverse accessions. It's now about, I think, 120 accessions. And we checked this viability, the viability after 2, 4, 8, 16, 32, 64, 128 years, which are with a sample size of 30 shoe tips. And this is how in parallel we monitor the viability, because if you can take out vials from your collection and check the viability, but then you reduce your stock, and at some point you would have to cryopreserve them again. No? So you have to be very careful to do this at a certain time point. No, surely this will be done at some point. No, we are thinking about after 10 years, perhaps after 20 years, but it depends. No, how many vials you have in your cryobank, and yeah. Bart, do you want to add something on this or on the second question?
I think you're quite right. And so we have regenerated also material which was in the cryo tank for 20 years, 25 years, and it behaved like it was only in the nitrogen tank for 1 hour. Yeah. Of course, it's difficult to prove that. I mean, but yeah. I think if I would not believe that cryopreservation would be something for the long term and that's quite stable, I probably would not do it, because there is so much investment in that, and this is, yeah. So and again, we can also refer back to the medical science and animal science, where I think recently there was a report that they recovered frozen bull sperm after being cryopreserved for 60 or 70 years, and it behaved like it was only in the cryo tank for 5 minutes. So yeah, that's yeah. I think it's okay.
Okay. Thank you, Bart.
It's
Thank
quite
you,
fine.
Rainer.
Thank you very much. Yeah.
Yeah. We have Elizabeth who raised her hand before we go on the chat where we still have one question to respond to. Elizabeth, please go ahead.
Thank you so much. This question is, I think, specifically for Bart, because Bart is working on bananas. Bart, we have TR4 raging through, and in the Caribbean especially, we've got, you know, certain types of bananas that we are really, really scared of losing. Are there any efforts to cryopreserve cell cultures of favorite banana species until we can CRISPR Cas9 in the resistance into all of them? Is that ongoing effort?
That's a good question. Yeah, that's because you're coming on to my other field, CRISPR-Cas. So indeed, so what we do is we now conserve the widest possible diversity. Yeah? So all the accessions that we are storing in vitro, irrespective of whether they contain they have TR4 resistance or other diseases, we store everything. So the widest possible genetic diversity. But related to your question, what's happening with so is there research done on that? Yes, so there are some initiatives involving classical breeding of bananas, but this is of course difficult because you have to rely on the wild ancestors from Southeast Asia, and then you have to make sure that you can create a parthenocarpic fruit, which is not easy, and a fruit that is nice to eat. But here in Leuven, we are collaborating with the university. We are indeed working on the CRISPR-Cas technology, and we recently have published a paper on DNA-free gene editing. So this is still in the pipeline, but we have to admit indeed that still there is not really a ready solution to replace the Cavendish, which is sensitive to TR4, and yeah, but we are working on that. But yeah, it can still take some time. And the thing is, okay, there are some bananas which are more resistant towards TR4 than others. But then, of course, then is the question: will people? Can they be commercialized? Will people like it? Can they be transported like they do now with the commercial bananas? So, yeah, there's still quite some question remaining. Yeah, but if you need more info on that, you can always contact me.
Yes, definitely, because we do have some precious bananas in the Caribbean that we would like to keep after TR4 goes through. So,
sure.
Thank you so much.
Okay, thank you. So we have two questions in the chat. One from Monsieur Ali is saying that what is the cost of cryoconservation for one accession per year?
Yeah, depending on the crop and where the work is executed. Some parts of the world staff costs are more expensive than other parts of the world. But for banana, we can say that one accession, the cost to cryopreserve one accession is around $1,500 and can go up to 2,000. Of course, you have to consider this as a long-term investment. So this means once it is cryopreserved, you have done the investment, then the only extra investment to store that accession per year is a few cents or maybe $1 or something, because it's the cost of liquid nitrogen. But the initial investment, and this is something I think good to realize, is not nothing. Yeah. So, but in case of potato, what is there? Maybe Rainer can respond to that.
Yeah, because of the region, it's lower. It's about the initial cost is about $500 per accession, and then the maintenance cost year by year is about $7. That what Bart mentioned. It's really important because this has to do with how you dimension your tank. You know? If let's say you have a tank that costs $40,000 but can hold 2,000 accessions, so the cost per accessions, the one-off cost, is very few. But if you have a huge tank and you don't have accessions inside this tank, so very few accessions, then this cost goes up. So if you start cryopreserving or start to build up a cryopreservation program, it may not be the best option to think in a huge, super fancy tank, but look for a more medium-sized tank that's more realistic to your throughput rate and the collection that you want to cryopreserve. Otherwise, yeah, you may take the investment may not be good.
Okay, thank you very much for both of you. So the last question we had from Yanelis Castilla from INCA. Dr. Rainer mentioned that the droplet vitrification is the most used method in gem plasma banks. Does it mean that is in general better results are obtained with this method?
Well, no.
Okay.
It's a good method. It's very practical. It's cost efficient. You get a high throughput rate. It has shown to be successful in many species and crops, but there are other methods that also work. Bart has shown many of them, and yeah, there's no golden protocol, golden method, I think.
But I think it would be a good start if you start with cryopreservation. It's a simple method. You do not need these cryoplates, and I think it showed that its value for many crops. So if you are starting from scratch, it's a good method to start with, I think, and then you can still see later on whether you have to apply other methods. But it's a good method to start. At least this is my opinion. Yeah.
Yeah, and for us, also our protocol, for example, for potato is based on the work of Bart on banana, no, and as it's the droplet method, and it has shown to be successful also in sweet potato and in oyuko and yacon and other crops, and also I think for cassava and yam, it's the method to go, as it has shown to be very successful and with high recovery rates.
Okay, thank you very much. So we have another question from Rodriguez from Cuba. He said, thank you very much for your presentations. Relative to initial explant, are seed a good material to be used in cryoconservation?
Okay, so a big collection. So you know, everybody has seen maybe the huge seed collections of Ilri, of Erie. I mean, if you have to store all that material in a cryo tank, yeah, then you need a lot of cryo tanks, and that can become a bit expensive, I think. But as I mentioned, maybe a core collection, I think that could be a nice thing to do. And for orthodox seed, cryopreservation is very straightforward. Eh? If something can survive -20 for 100 years, probably these seeds can also be just plunged in liquid nitrogen and be regenerated afterwards. So for orthodox seeds, only, I think, for a core collection. And for recalcitrant seed, yes, that might be the only option if they cannot be stored at -20. Yeah.
Okay, thank you. So you still have a hands up? Oh my goodness. We are beyond the time, so I will just take these 2 questions, please. Then we will stop the webinar. So Ahmad, you can open your mic.
Yeah, thank you so much for allowing me to ask another question. As you mentioned that the initial cost of establishing cryopreservation unit is a bit higher on a bit higher note, from starting from $500 to $1,000 per sample or per accession, and depending on region and all that. My question is, what do you think? At what stage one should think that they should preserve their samples using this technology?
At any stage, you as it's just the future. No, it's the now and the future. If it depends how okay, for the first year it will cost you more money, no, but then you will save money. It's for the long-term conservation. If you want to conserve something on the short or mid-term, your cryopreservation might not be the option to go. But if you're thinking about 10, 20 years or bejoin this, surely cryopreservation is the most cost-efficient way to conserve your collection.
Okay, thank you. Elizabeth, please, can you open your unmute and raise your question? Thank you.
Thank you, Marino, for allowing us to ask these questions. I want to go to the commercial application that you mentioned, Rainer. Are there companies that's doing that at this point in time? For us in the Caribbean, being able to conserve or cryopreserve sweet potato virus-free for rapid regeneration is very attractive. So what and who is doing the commercial applications at this time? Thank you.
There are no commercial applications. They're doing cryopreservation of sweet potato is at SIP. Bart has worked on it and also published on it. I think from Korea, South Korea, I think there are some publication from Japan, but there's very few institutes worldwide working on cryopreservation of sweet potato. And what I mentioned, the commercial, it's not so commercial application. It's your capacity to react very quickly when there's a disaster. No, that you have a lot of plants available in a very short time period, but you don't know when the disaster happens, so you can't really prepare through maintaining huge in vitro collections for this. I see there's a new opportunity for cryopreservation as an application, bejoined cryotherapy and other things. No, yeah, but there's no commercial company out there, as long as I know.
All right. No, thank you very much. And that's also important for us in the hurricane belt in the Caribbean. We have hurricanes now that can flatten an island. So yeah, it's certainly attractive. Thank you so much. Thank you all.
Okay. So thank you so much, Dr. Rainer and Dr. Bart, for your excellent presentation. So ladies and gentlemen, we are now at the end of this webinar today. So on behalf of the organizing team, I would like to sincerely thank you for joining us today. We look forward to seeing you again tomorrow at 1 p.m. European time. So we continue with the issue related to cryoconservation, cryopreservation. So I wish you a very nice day, evening or afternoon, wherever you are. And then please come back tomorrow and on time. Thank you. Goodbye.
Bye-bye.